Hold on.
Me too.
Started what?
I think so.
Alrighty, here we go.
I did.
Yeah, I didn't play soccer.
Yeah. Crazy.
We did that yesterday.
Okay.
Okay, we're learning about meiosis. I'm going to do the whole process again. You ready for
It
this?
took me forever.
Erica, do you think you could sit right there?
Yeah.
Free.
Here, take this seat.
Or there? Yeah, that's even better. Because I want to stand to this side.
Yeah. Okay.
If I'm standing over there, lots more people. Yay. Thank you. Okay, so we're doing the whole process of meiosis from the start. Do you remember the phases right here? Prophase One, Metaphase One, Anaphase One, Telophase One. And then enter Kinesis. And then Prophase 2, Metaphase 2, Anaphase 2, and Telophase 2. After we go through both of those processes, you won't have a diploid cell anymore. You know what you'll have?
Sperm cell.
Which is called a, not a
Haploid.
diploid, a haploid cell. Haploid cell. A haploid cell is also called N because it has one set of chromosomes. The N means one set. 2N means two sets. We're going to go to cells that have one set of chromosomes. Are you ready? It's exciting, isn't it? All right, here's a big gonadal cell right here. Imagine this is a cell in a man's testes. A giant testes cell. Think how big the guy would be if the cell was that big. He'd be a giant. He'd go all the way to the moon. Okay, so anyway, but what we do is we start in Prophase One. Now, Prophase One is kind of like in mitosis. The nuclear membrane dissolves. You remember what else happens? Just shout it out. Does anyone know?
Spindle fibers come out of the centromere.
The centrosomes move to opposite sides of the cell and spindle fibers come out. I'm just going to draw the spindle fibers kind of small. Just imagine they go all the way across. And the spindle fibers attach to the chromosomes. And the chromosomes, they line up in the cell. But this is where something different happens from mitosis. They find their homologous pair. We call these things homologous pairs. The paternal chromosome has a set, the maternal chromosome has a set, and those chromosomes that match find each other. 10 points. If you could tell me this is called a homologous pair, but it's got two other names. 10 points if you can tell me, 5 points each, the two other names of this pair of chromosomes.
I got one.
Nick Frazier got it quick. Right on both for orange.
Wait, did you say so?
One right for green. You got two more names. Both right for blue.
Nice. Good job.
Peach, both right. Anyone else?
Wait, if we got one, can we get another?
One, yeah, one is fine. Get 5 points for one.
Can we get another one if you want?
No, I need the one that you have.
I just recording.
And then you recorded it going back
No?
I feel bad for whoever watches this.
You can turn.
Okay,
Did you
now
already
it's
turn one in?
Yeah.
And you have the other one there? Bring it here. White, not right.
How you doing?
Green, got the other one. Oh, thank you. No one else? Okay, so this is called This is called a homologous pair. It's also called a tetrad because it's got 4 sister chromatids. It's also called a bivalent. Just got 3 names, just like I have 3 names. Mr. Willis, Tom Willis, and
Pro.
DJ Grandmaster T. Or Pro. Okay, so anyway. They form these tetrads. And what happens when they form these tetrads?
Go to the center.
If something happens,
They swap. They cross.
they can cross over pieces. Pieces can get switched from one to the other. That's a big deal. Why is that a big deal? To switch pieces of chromosomes. It makes for new chromosomes that have never existed before, and when you pass them on to your children, they will get traits that are combinations of your chromosomes, and they'll be very different from you. Or any of their brothers and sisters, because every time you do this, the pieces that switch are different. So that one time these 2 pieces might switch, but another time those 2 pieces might switch, and the next time you make sperm cells, those 2 pieces might switch. So each kid, each sperm cell is going to have differences in it. This is how you get variation. This is why all kids don't look alike. That's very important to have all kids not looking exactly the same.
Well, when identical twins, what would that be?
Okay, so identical twins are formed from after all this happens, and you have a sperm cell and it meets the egg, and then you have a zygote, which is going to grow into a human, and for some reason, that cell divides in 2 and grows into 2 humans with the same DNA. So that's identical twins, which is a little bit, it's not involved in this process. It's after this process. Okay? How are we doing so far? That was all Prophase 1. Prophase 1 gets you to here. Okay? Thomas, you with me over there?
Yes, sir.
Sweet. Now, Metaphase 1. What happens in Metaphase 1?
Independent assortment.
Huh?
Chromosomes are pulled to the center.
Yeah, but what do the chromosomes do?
Line up.
They like vary, like variation and stuff.
In Metaphase, what's Metaphase? Metaphase, starts
Middle.
with an M? Middle. They go to the middle. In Metaphase, they line up in the middle. Now, some people were saying independent assortment happens here. That's true. Independent assortment is the idea that they could all be green on one side, but maybe the next time you make a sperm cell, you could have 2 greens and a white. Maybe the next time you make a sperm cell, it could line up like this, where you have 2 whites and a green. In a human cell, there's 23 pairs of chromosomes, so there's many, many ways that they can line up. Again, this adds to the variation that's found in the species. You get every time you make a sperm cell, they line up a different way. So that leads to a bunch of different sperm cells. And that'll make all the kids different. We're adding to variation here. Okay, that was Metaphase 1. What's the next phase?
Anaphase.
Anaphase.
Anaphase 1, the chromosomes separate and move to opposite sides of the cell. But I want you to notice that it's not breaking apart in the middle like in mitosis. It's each member of the homologous pair is separating and moving to opposite sides. And so now we have a cell that is going to split into 2 cells, and you can see we're reducing the chromosome number by half. We no longer have 2 sets of chromosomes on each side. We're moving toward a haploid situation where we just have 1 set of chromosomes on each side. Y'all okay? Telophase 1. The chromosomes arrive at opposite ends. These spindle fibers break down. Spindle fibers break down. And a new nuclear membrane forms. New nuclear membrane forms. And the cell starts to pinch inward. Cell starts to pinch inward. We're almost to 2 new cells here. And that's telophase. 10 points if you and your partner can tell me what happens to these chromosomes in this phase, Telophase One. 10 points! Ten points if you can tell
Do
me.
they like split?
What happens to the chromosomes in this phase?
Oh, my game.
I got you. I got you that. I saved you.
Oh, really?
I'm watching.
You announcing the game tonight?
No, I'm out of town.
Who's gonna do it?
No, you know who's doing it? Mr. Yarborough.
Is Ryan not gonna be up there?
That's good.
I'm not sure. White. Not right. Blue. Correct. Peach. Correct. Orange. Correct. Purple. Not right. Green. Correct. Anyone else? Did I get everybody? So what these things do in telophase, if you don't know this, you need to know this. This is part of your essay. What these things do in telophase is they uncoil back into chromatin. Remember, these are coiled up chromosomes. They'll uncoil back into chromatin. That always happens in telophase, whether it's mitosis or meiosis. And now we have the cell splits in two. Just yell out. What's it called when the cell splits in two? Cytokinesis. Do it, Thomas. Cytokinesis. And now we have 2 cells. Isabel, count how many chromosomes are in this cell.
3.
Yes. And do you know how many we started with? When the original cell? 6. So do we have half the number of chromosomes?
Yes.
That's haploid. Haploid means half the number of chromosomes. Does this make sense? Are y'all with me? Are we done with meiosis?
No.
Have we made sperm cells yet? We have not. Because what we have to do now is divide up these sister chromatids that are still stuck together. We've got to divide them up. There's no reason to have 2 copies of information. This will break apart. And that's what happens in meiosis 2. So let's go through meiosis 2. There's this brief rest period called interkinesis before meiosis 2 starts. And what happens in interkinesis? Well, one of the things that happens is the centrosomes divide and form new centrosomes. And that's what happens over here too. These centrosomes are copied. There are enzymes that pull that off inside the cell. And then we go into prophase 2. And by the way, meiosis 2 is just like mitosis. Every step's exactly the same. Lachlan, do you remember mitosis? Well, it's the same thing in meiosis 2. So you already know this a bit. Isn't that great? Here we go. Prophase 2, the centrosomes move to opposite sides of the cell. And this happens on both sides. Centrosomes move to opposite sides of the cell. And we can put it up here for you if you want to see. The nuclear membrane dissolves. What else happened in prophase? Do y'all remember?
Turn the chromatin turns to chromosome.
The chromatin coils up into chromosomes so we can see them. Very good, Nick. Frasia. What else happens? Prophase. Come on, y'all know.
They shoot out spindle fibers.
Yes, do it, Isabel. They shoot out spindle fibers. And what do the spindle fibers do?
I don't know.
They hold them in place.
They hold what in place?
The
Chromosomes.
So are you saying the spindle fibers connect to the chromosomes?
Yes.
Yes, spindle fibers connect to the chromosomes, and that's prophase 2. What happens in metaphase 2?
They line up in the middle.
Yes, they line up in the middle. Whoop. All lined up. And these over here, all lined up. That's metaphase 2. Where's Anna? Anna, can you tell us what happens in anaphase 2?
Split up.
Yeah, they split up this time at the centromere. We got to split up those sister chromatids and get them to opposite sides. Split them up on both sides. Split them up. Split them up. Do it, Anna. That's your favorite phase? Nick, what's up?
What are the individual strands of the chromosomes called again?
Sister chromatids. They're called sister chromatids, but Nick, when they break apart like this, now they're called daughter chromosomes. So sister chromatids, daughter chromosomes. And what would you call, Nick, the whole thing here? Do you remember that?
Are duplicated chromosomes?
Yes! Nick, you're on top of your game. I respect that. Here we go. This breaks apart and goes to opposite sides. Are y'all with me so far? G-Ray, you good back there? Now, we go to telophase 2. We're about to have sperm cells, y'all. Telophase 2, these chromosomes arrive at opposite ends. These spindle fibers disappear. A new nuclear membrane forms. And what happens to the chromosomes? Just yell it out.
They uncoil.
They uncoil. Very good. So that's going to happen in all of these. Chromosomes reach the end. Spindle fibers disappear. New nuclear membrane forms. Chromosomes uncoil. Spindle fibers reach the end. Chromosomes reach the end. Spindle fibers disappear. New nuclear membrane forms. Chromosomes uncoil. Chromosomes reach the end. Spindle fibers disappear. New nuclear membrane forms. Chromosomes uncoil. And now the cells start pinching inward. They start pinching inward like this. Pinching inward like this. It's going to happen on both sides. Here we go. And it'll finally break apart in cytokinesis. And guess what we have now?
Sperm cell.
4 sperm cells. These will grow a tail.
What is that tail called?
The flagellum. And it's actually really long. The flagellum is like 20 times longer than the head of the sperm. It might be more than that. I don't know, 100 times. I'm not sure. Somebody Google that. Anyway, the tail of the sperm is a lot bigger than the head of the sperm. But anyway, the tail grows, and now this thing can fertilize an egg. And I want you to notice something about the sperm cells. They're all different. They're all different. This has 2 greens and a white. This has 2 whites and a green. This has 2 whites and a green with a piece of white. This has 2 greens and a white with a piece of green. They're all different. Every time you do meiosis, the sperm cells are different. And so the reason why you look like you do is because a certain sperm cell fertilized that egg and put certain genes in there that made you look the way you do. If a different sperm cell would have gotten the egg, you would have looked totally different. You could have been a guy instead of a girl instead of a guy. You could have been taller, you could have been shorter, you could have been stronger, you could have been weaker. You could have had a genetic disorder like Down syndrome if that other sperm had made it into the egg, but it didn't. The one that fertilized the egg was the one that made you. And usually, when people are having sex, shh, there's about 600 million sperm cells that are ejected into the female that are all trying to get to that egg. And you know how many get there? One. Well, about 100 get there, but only one fertilizes the egg. So you had about a 1 in 600 million chance of turning out the way you did. You've already beat the odds. Isn't that interesting? Wow. I could have been real tall. Probably not, because both my parents are short. So almost every one of their sperm cells had chromosomes with short genes on them. So I wasn't going to be tall. Oh well. I got the rest of it. Great looks, big muscles, perfect physique, and that's what it is. I'll take it. Okay? Are we good? That's meiosis. Yay! You want to see video footage? Video footage of meiosis. Here we go.
In Prophase One, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses and they exchange.
We're starting over.
In Prophase One, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses and they exchange segments by crossing over. By Metaphase One, the nuclear membrane has disappeared and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In Anaphase One, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
Yay! Doesn't that look like kind of like what I have on the board? Especially if you look at it right there. Only I had 3 chromosomes in my model and they only have 2. And how many would a human cell have? 10 points! If you can tell me how many chromosomes would a human cell have in it at this point
Would it be 46
at the end of
times 4?
telophase 2? 10 points if you can tell me.
Oh, yeah, because... Okay,
Wait, what phase is that?
we're just going to go with it.
At the end of telophase 2, when you get to this phase, how many chromosomes would a human cell have in it? Orange correct. Orange, you glad I didn't say banana? Peach incorrect.
It's okay. What?
Green incorrect. Purple correct.
Let's go with that. Let's go with it. Let's go with that.
Anyone else? White correct.
What was the issue?
Blue incorrect. Okay, listen up. It would have, all of you that missed it, 3 groups missed it, so pay attention here, it would have 23. Because it starts, the diploid number is 46. We start with 46 at the beginning, but when we go through all of this, we divide the chromosome number in half. So we started with 6 chromosomes, and now each one of them has 3. So humans, you start with 46, and you end up with half 23. So these are haploid cells. So this sperm that has 23 fertilizes an egg that has 23, and actually you end up with a new cell that has 46. And that can grow into a human. Yeah.
So once we're done with meiosis 2, those daughter chromosomes, they turn into chromatin.
Yes.
And if the sperm needed to divide again,
The sperm doesn't divide again.
just in general for cells,
Okay.
if it needs to divide again, would the daughter chromosomes turn into chromosomes after they preform in prophase?
If this cell could divide, it would have to go through prophase, metaphase, anaphase, telophase. The daughter chromosomes would, these chromosomes would coil up again. But you would have to copy the DNA before you could divide it. And the sperm never do that. Sperm will never divide again. Their only mission is to find an egg.
That answers my question.
Yeah, so it doesn't happen with these cells. These cells are special in that they don't divide anymore. They have to find another haploid cell. That's the only purpose for these cells, to try and make babies. Make sense? We good? Okay, could you turn that off for me?